doctor, I'm Tian Zhang. I chose the first journal, so now I will explain the development of surface-enhanced Raman scattering-based surface corrugated nanopillars for biomolecular detection of colorectal cancer. Now, let me give a brief introduction of colorectal cancer. Colorectal cancer is a type of cancer that varies widely around the world. The recovery rate is high following surgery treatment in the early stages, but it dramatically decreases in the third and fourth stages. Therefore, early diagnosis and treatment are crucial to increase survival rate among CRC patients. The current method of CRC early stage diagnosis relies on symptoms or fecal occult blood testing in the majority of patients. However, the sampling is very inconvenient, which reduces suspected patient motivation. Furthermore, CRC is inhomogeneous and it has complex gene expression subtypes. Medical professionals have considered molecular subtypes of CRC for personalized medicine and optimal treatment. As a result, scientists can provide further insight in terms of molecular information for developing new diagnostic approaches. MicroRNA, which are small non-coding RNA subsequences containing 18 to 24 nucleotides, are transported by exosomes into bodily fluids as post-transcriptional regulators of gene expression. There is a growing body of evidence showing that exosomal microRNAs have a strong correlation with the occurrence and development of various types of tumors. Those findings have attracted increased attention to microRNAs as ideal markers for early diagnosis, particularly in CRC. However, the detection of exosomal microRNA molecules is still considered to be a challenging work. Surface-enhanced Raman scattering spectroscopy is a powerful analytical tool for microRNA detection. SERS is carried out when nanostructured surfaces of noble metals absorb irradiation with light. The energies are transferred to localized surface plasma resonance with near substrate, and other signals are enhanced. The current key is nanogaps among the metals generating strong SERS effect known as hotspots. However, the inhomogeneous aggregation of nanoparticles or the imbalanced distribution of hotspots may result in irreproducible measurement. To address this problem, modified ordered nanostructures on solid surface by mature lithography fabrications have been developed, such as nanoballs, nanoholes, and nanopillars. The gaps between the adjacent nanostructures produce SERS enhancement due to strong electromagnetic fields and plasma coupling effect. In the previous study, we found that SERS enhancement on etched nanospheres were dominated by sub-10 nanometer pitches and high surface roughness instead of particle size. The surface roughness and sub-10 nanometer nanocorrugations or polysurgery beads were also quantified to provide 2 orders of magnitude of SERS enhancement. As a result, in the current study, not only the aspect ratio of nanostructures, but also the surface roughness of silicon nanopillars were adjusted to further improve the SERS effectiveness and demonstrated the fabrication process of silicon nanopillars with the nanoshielding mechanism, but we also increased surface roughness to markedly improve the Raman signals. Not having secondary modifications and the chemical synthesis of nanoparticles may reduce complicated procedures and provide benefits for mass production. We further combined SERS active silicon nanopillars with sensitive molecular probes, which can immobilize onto pillar surfaces to obtain a fingerprint spectrum for macro R21 recognition and detection, as shown in Figure 1. These characteristics are very beneficial for highly sensitive and stable detection of RNA molecules. Next is the material and method part, and these are the materials that we need in this study. Below that is oligonucleotides and how we employ a fluorescence screening SERS enhancement design to test microRNA-21. And this slide shows the fabrication of nanopillar structures by nanoshielding methods. Figure 2 stands for the fabrication process of nanopillar array. Then we need to prepare and immobilize the probe. Next is the characterization of nanopillar chips with scanning electron microscopy and atomic force microscopy. A high-resolution thermal field emission scanning electron microscope was employed to image the structures of corrugated nanopillar arrays. Fabrication under different etching conditions. The SEM results are shown in Figure 3. The surface roughness was further investigated with atomic force microscopy. To understand the contributions of SERS enhancement, we independently analyzed macro ranges, nano ranges of roughness of the silicon nanopillars. And Figure 4 stands for the corrugated nanopillar structure for SERS enhancement. And these are the SERS measurements. SERS spectra were acquired by a macro-Raman spectrometer. Raw SERS spectra were processed through LabSpec application software for smoothing the data and adjusting the baseline before further analysis. Furthermore, to analyze the detection limits, series concentrations of R6G were placed on the silicon nanopillars and Raman signals were recorded in Figure 5. For microRNA-21 detection, SERS spectra were acquired by a portable Raman spectrometer. Figure 6 stands for the Raman spectra of microRNA-21 detection with silicon nanopillars. To test the specificity of this sensor, microRNA-21 similar sequences with 2, 4, and 6 mismatched bases were used to complete with probe 21Q binding sites. Now is the result and discussion part. First is the fabrication of nanopillar binosaurs. Silicon nanopillars morphologies were analyzed by SEM and macro-range and nano-range roughness by AFM. Figure 3 shows the top view and cross-section view of silicon nanopillars by SEM. Note that pillar structures did not form within etching times shorter than 40 seconds in all experiments. After optimization, the structure with 160 seconds etching time provides a more uniform and repeatable nanopillar structure. Moreover, the surface roughness of macro-range structure and nano-range structures on the pillar sides were measured by AFM as shown in Figure 4. These findings align with the methodologies in the SEM images. Furthermore, SERI signals were measured from 5 fabricated samples. We found the relationship between Rm and Rn with Pearson's correlation. Second is SERS signal enhancement by corrugated nanopillar banosol. To detect the SERS enhancement on the series of nanopillar structures, we compared the Raman intensity of a Baldwin 6G dye. Typical Raman spectra of R6G were well resolved with these peaks. We further calibrated series concentrations of R6G. The SERS signals were measured from 5 of the surfaces at 160 seconds, as shown in Figure 5. The limit of detection of R6G was estimated to be the 8th power of 10 mm based on 3-time measurement of black signals. The enhancement factor was calculated with equation. In our experiment, INRS was obtained from 0.01 m of R6G solution with bare silicon. Its average intensity was 3.5 counts. CSERS was 10 nm. The EF of silicon nanopillar was obtained close to 1 plus the 7th power of 10. And third part is the macroRNA 21 detection on corrugated nanopillar banosols. The hybridization process between the target molecule macroRNA 21 CY5 and probe 21Q could easily be distinguished through Raman spectrum variations, as shown in Figure 6. Without tripling macroRNA 21 CY5, the Raman signals for probe 21Q conjugated on the gold surface represented a small broadband spectrum. These signals were used as a negative control in our system, representing probes catching no target in the environment. In summary, a nanobiosensor with surface-enhanced Raman scattering capability has been introduced for highly sensitive macroRNA detection in this study. The fabricated nanopillar structures can effectively extend hotspot distribution into the third dimension, providing considerable enhancement in Raman signals. More than 3 orders greater than nano-optimized gold deposited silicon nanopillars. This nano-biosensor can serve as an SERI solid substrate for macroRNA 21 recognition in a rapid and reproducible manner. And the target molecule macroRNA 21 can be easily monitored through a Raman spectrum variation with a PCR comparable concentration at 100 pm. The proposed silicon nanopillar sensors achieve good selectivity to 2 base mismatched macroRNA. Furthermore, without the sequence amplification process, the whole testing can be completed in 1 hour. Moreover, due to their easier integration, nanopillar arrays may be employed in a microfluidic platforms with lateral flow or automatic RNA sensing. These compact, comparable biosensors are also suitable for emerging point-of-care applications, such as pesticide poisoning, drug testing, and toxic chemical analysis. And these are the references. That's all. Thank you so much for your listening.